Hi everyone, welcome to the channel Exam Ignite. So this is the continuation of the brand new series of subject-wise and module-wise preparation for VTU exam. And today we will be going to study about Module 5, Trends in Bioengineering. The first topic, before going to the topic, the Module 1, Module 2, Module 3, Module 4 are already uploaded in the channel. Check the description box for the link. The first concept is muscular and skeletal systems as scaffolds, where you'll be learning architecture and mechanism, bioengineering solution for muscular dystrophy and osteoporosis, then bioprinting techniques and materials, 3D printing of ear, bone, and skin, 3D printed food, electrical tongue and electrical nose in food science, DNA origami and biocomputing, bioimaging and AI for disease diagnosis, self-healing bioconcrete, then bioremediation and biomining via microbial surface adsorption, then removal of the heavy metals like lead, cadmium, mercury, and arsenic. First question, explain in detail the architecture of the muscle with a neat diagram. Muscle are the contractile tissue that is responsible for the movement, posture, force generation in the human body. The architecture refers to the muscle fibers, fascicles, and connective tissue are organized. This is the muscle architecture. Muscle, it is surrounded by the epimysium, outermost contractive tissue. Then fascicles, it's a bundle of muscle fibers surrounded by perimysium. Muscle fibers are cells, the long cylindrical cells, multinucleated, surrounded by endomysium. Myofibrils, it is composed of the repeated units called sarcomeres. This contain actin has thin and myosin has thick filaments. Sarcomere, defined between the two Z lines, contains A band, that is dark myosin plus actin overlap. I band, which is light, only actin. H zone is only myosin. We are having different types of muscle architecture with their examples. Next question, elaborate the different engineering solutions for muscular dystrophy. Muscular dystrophy refers to the group of genetic disorders causing the progressive weakness and degeneration of the skeletal muscles due to the absence of defect and dystrophin protein. What are the engineering solution is firstly gene therapy, where we insert a functional dystrophin gene into the muscle. Second one is CRISPR. It is a gene editing where edit the patient's own gene to restore the or to skip the faulty exons. Then stem cell therapy. Here transplantation of the heavy muscular progesterone or cells. Protein replacement therapy where administering synthetic or recombinant dystrophin-like protein to compensate for defective one. Next, biomaterial scaffolds and 3D printing where engineered muscle tissues using biodegradable scaffolds. Drug-based therapy where we use steroids to slow the degeneration. Osteoporosis is a skeletal disorder that is characterized by reduced bone mass and the myoarchitectural deterioration leading to the fragile bones and increased fracture risk. So, what is the normal and in what is in the osteoporosis, the difference is given, please to refer. Bioengineering solutions are bone grafting and scaffold, where these are seeded into the osteoblast to regenerate the bone. Uses the calcium phosphate or hydrogel composites. Next, stem cell therapy here, MSCs enhance the bone repair, can differentiate into osteoblast. Nanotechnology here, nano carriers for the targeted drug delivery, improves the drug absorption and reduces the side effects. Tissue engineering, it combines the biomaterial and growth factor and cells. It builds the 3D bone graft mimicking the native bone. Biochemical treatment, drugs are used. Next, in 3D printing for implants, personalized bone implants using the CT scan or 3D printing. Difference between 3D printer and bioprint. 3D printer is a mechanical device that creates 3D print objects layer by layer using the materials like plastic or metal. But bioprinting, it's a specialized 3D printing that prints the living cell, or you can say bioinks and biomaterials to fabricate the biological tissue or organ. This comparison table is very important. Materials here in the 3D printer are plastic PLA or ABS metal resin. In the bioprinting, living cells, that is stem cell, hydrogels, bioinks. Here the purpose is for the industrial design prototyping tools. Here tissue engineering, regenerative medicine. So what will be the resolution in 3D printer? Higher mechanical precision. Here it is optimized for the biological resolution. What is the nozzle type? Here it is thermoplastic nozzle. Here it is pneumatic, that is piezoelectric or micro extension head. Temperature, here it uses to melt material, but here it requires ambient or body like temperature. Sterility requirement is not required in the 3D printer, but it must operate in the bioprinter. Examples, here you can give gear, models or spare parts. Here it is skin, ear, cartilage or muscular tissue. Next question, write a short note on bioprinting techniques and materials. Important question. Bioprinting is a type of 3D printing that uses biomaterials and the living cells to fabricate the tissue-like structure that mimics the natural tissue and organs. It is a key technology in tissue engineering and the regenerative medicine. So this technique has 3 stages: pre-bioprinting, bioprinting, and post-bioprinting. So first technique is inject bioprinting. Here works like a normal inkjet printer. It uses thermal or piezoelectric forces to deposit the bio ink droplets. Advantage is high speed, low cost, good resolution. Limitation is not ideal for the viscous biolinks or constructs. Extrusion-based bioprinting, here it dispenses continuous filament of bioink using the pneumatic or the mechanical pressure. Compatible with hydrogel, self-spirites, and biomaterials. Advantages: it is suitable for high viscosity material and large scale constructs. Limitation: shear gear may reduce cell availability. Laser based printing, here it uses focused laser energy to transfer the bioink droplets onto the substrate. High resolution and nozzle free, reducing the clogging. So all these 3 are given here. This is the inkjet bioprinting, this is the laser assisted bioprinting, this is the extrusion bioprinting. Now last one is SLA bioprinting. Here it uses a visible UV light to cure the light sensitive bioresins. Offers excellent spatial resolution. Limitation is limited to photopolymerizable material. This is the figure. Next, materials. Natural polymers, we use collagen, gelatin, then fibrin, hydraulic acid. Advantages: biocompatibility mimics the ECM, supports cell adhesion. Limitation is weak mechanical strength. Synthetic polymers, example is PEG, PLA, PCl. And advantages is tunable mechanical properties and stable. Limitation is poor bioactivity. May need bleeding with natural polymers. Then comes decellularized ECM, here extracted from tissues or organs with all cells removed, maintains the native biochemical cues. Then cell aggregates and spheroids, here used in scaffold-free bioprinting, high density of cells, better tissue integration. Discuss the materials used for 3D printing of human ear. So 3D printing of human ear, it replicates the anatomical structure, flexibility, and the function using the biocompatible material. It combines the scaffolds and the living cells to form an ear-like construct. Material we use is hydrogel-based bioink, that is collagen, gelatin, and alginate. It mimics the ECM of our ear cartilage. It supports the cell growth and biodegradable. Cartilage cell, we use chondrocytes. It is taken from the patient to the donor, mixed with the hydrogels to form living structures that grow in cartilage. Synthetic polymers, we use PLGA, PCl, and PEG. Stem cells, this enhance the tissue generation and healing. Support gel, we use a fresh named gel. So it is a gelatin slurry used during the printing to support the soft materials as the ear forms. Processes: first we take the image, that is capture the patient's ear structure via SCT or MRI. Design a CAD model of a ear is created. Material preparation is scaffold plus bioink with cells. Printing layer by layer deposition, culture and maturation in bioreactor for the tissue formation. This is the diagram. First we extract the model. Model is processed, model mirroring, and we have a Boolean operation. From that, this is a CAD designing. After that, in the manufacturing, it gets manufactured, and this is a customized model. Important points you can refer. Then discuss the materials used for 3D printing of human bone. 3D printing of human bone aims to fabricate the bone-like structures that replicate the mechanical strength, porosity, and the biological properties of natural bone. This is achieved by combining biocompatible scaffolds and the living bone-forming cells for applications in orthopedic, dental, and craniofacial reconstructions. Key materials are ceramics, calcium phosphate paste, synthetic polymers, PCl, PLA, PLGA, natural bioinks, hydrogels, composite materials, we combine HA plus PCl or PLA plus TCP. So this is how X-ray or the broken one is taken, the image, then we process it in the V design, we process it, and finally fabricated bone scaffold, then we get the bone. That is, imaging uses CT or MRI to scan the defect or the bone structure. It generates a CAD model of bone and porosity. Material preparation is done here, and polymer scaffolds with the cells or bioinks. Printing using extrusion material or bioprinting. Post-process mature scaffolds in the bioreactors with osteogenic media. Discuss the 3D printing of human skin. So basically, it is a biological process where fabricate skin-like structure using the biocompatible materials and living cells. So it is applied in the burn treatment, wound healing, drug testing, and cosmetic research. It's a multi-layer of natural skin. Materials we use is collagen, gelatin, fibrin, hyaluronic acid, and alginate. Cells we use are four types of cells. Synthetic polymers we use three types. Then stem cells we use a fresh gel. So then the skin is manufactured. So how imaging or design is first done, then material preparation is done, then cell mixing, next is layer by layer bioprinting, then post-processing, that is culturing in bioreactors. Explain the process or working principle of electric tongue and electric nose in food science. An important question. Electronic tongue, it is a multi-sensor system that detects the basic taste attributes, that is sweet, sour, or anything. Chemical properties in liquid or semi-liquid food samples. Sensor array, where multiple non-specific cross-section chemical sensors like lipid membranes, metal oxides interact with the taste molecules in the sample. Signal conversion, these interactions cause electrical changes, voltage, current, impedance in the sensors. Pattern recognition is done, then comparison. Application in food science for monitoring the saltness or sweetness in beverages, detecting the bitterness in pharmaceuticals, then ensuring the freshness and fermentation. Advantages, you can say, it is for the taste analysis, rapid testing, then for the sensitivity to detect the adulteration of the food or anything. Electric nose, it is an artificial olfactory system used to detect and identify the voltaic organic compounds responsible for the odor or aroma. Sampling system, where next is sensor array, we use metal oxide semiconductors, conducting polymers. Signal generation, VOCs cause changes in the electric resistance or frequency in sensors. Pattern recognition system, we use AIML algorithms. Display and analysis, results are interpreted and displayed as odor fingerprints. Application in the flavor profiling, spoilage of food detection, then in the geographical origin, brand identity. Advantages of electric nose, non-destructive testing, then early spoilage detection, automation, and the high throw output. Describe the concept of DNA origami and the biocomputing. So DNA origami is nanotechnology technique that folds a long single DNA strand into specific 2D or 3D shape with many short staple strands. The precise folding mimic the art of the paper origami but at the nanoscale. Okay, so these staple strands bind specific parts of the scaffold, causing it to self-assemble in the design shape structures and can be programmed with high accuracy. Note down the roles in biocomputing and some of the points. Next comes bioimaging and artificial intelligence in technique in disease diagnosis. So, firstly, MRI. MRI, what we do, we use magnetic fields and the radio waves to create the detailed soft tissue images. Detection of tumor, then brain disorders. CT scan, computed tomography, here X-ray of images from the multiple angles reconstruct the cross-section area, bone fractures and internal injuries. Ultra imaging, high frequency sound waves reflect off the tissues. Fetal imaging or organ assessment. PET scan, it is detects the metabolic activity using the radioactive traces, cancer diagnosis, brain disease. This is the image of an MRI. Next comes AI indices. So AI basically takes the data as input that is fed in the large database or the patient's record. Training, we train the models with the recognize the patterns linked with a specific disease. Then it predicts and classification where AI, it predicts diagnosis and classify the disease. So this improves the accuracy as more data becomes available. You can write that medical images sent through AI algorithm, pattern recognition, and diagnosis the output. Application in cancer detection, radiology, pathology, then predictive diagnostics. Describe the concept of self-healing bioconcrete and explain the role of bacillus spores, calcium lactate, nutrients, and biomineralization process in this technology. So, self-healing bioconcrete is a innovative constructive material that can automatically repair cracks and damage using the biological process. This technology improves the durability lifespan of the concrete structures by sealing cracks without human intervention. This concrete is prone to cracking, which reduces its strength and durability. Actually, concrete. So this self-healing it incorporates a bacteria, usually spores that remain dormant inside the concrete matrix. To the crack form, water enters. These bacteria get activated. They induce biomineralization, precipitating the minerals that fill this and seal the cracks naturally. Bacillus spores, these are dormant bacterial forms embedded into the concrete. Bacillus species are chosen because they are alkali resistant and survive in the harsh concrete environment. When cracks allow water and oxygen in, the spores germinate into active bacteria. They metabolize supplied nutrients to produce a mineral for crack repair. Then calcium lactate nutrient, what these do, they act as a food for the bacteria. It is added to the concrete mixture along with the spores. So when bacteria becomes activate, they consume this calcium lactate and produce a calcium carbonate. So this calcium carbonate precipitates and fills the crack. Then biomineralization process, this is a biological process where bacteria induce mineral precipitation. That is, bacteria metabolize calcium lactate, they produce CO2 and calcium ions, react to form the calcium carbonate crystals. These crystals act as a natural glue, that is sealing cracks and restoring the bioconcrete integrity. This process is environment friendly and extends the concrete life. Next question, explain the biomediation, remediation and biomining via microbial surface adsorption. So bioremediation is a biological process that uses microorganisms to remove, degrade or detoxify the pollutants from the contaminated environment like soil, water or air. So mechanism via microbial surfaces, some of the bacteria, fungi have the cell walls rich in functional groups, carboxyl, hydroxyl or ammonia. They bind to pollutants. Surface adsorption is a process where contaminants adhere to the microbial cell surface. This occurs through the biosorption, involving mechanisms like ion exchange, complexation, and the precipitation. Now, adsorbed pollutants become immobilized or transferred into less toxic form. Next comes biomining. It uses microbes to extract the valuable metals like gold, copper, uranium from the low-grade ores or mining waste by mobilizing metals through bioleaching or biosorption. What happens here is microorganisms attach to the mineral surface and adsorb the metal ions. Adsorbed the metals can be oxidized or reduced by the microbial enzymes, making them soluble. So metal dissolve in the solution and can be recovered through the convention extraction techniques. So common microbes are, few of them are given. Next application is the recovery of the copper and gold from the low grades, environmental friendly. They can be used in the mining waste treatment. That was all about the module 5. So module 1 to 5, everything is uploaded. Go through all the modules. I'll be uploading the important questions for the exam also. Go through that and prepare well for your exam. Do share, like, and subscribe my channel. Thank you.